All right, everybody, welcome back. A little bit of housekeeping. Because everybody has had so many great questions and because Dom and I have been on the stage a lot, we're going to axe our particular session to stay on track, and we'll plan to have a community call about therapeutic strategy at a later date so that we can continue having talks with the lovely people who have agreed to share their time and effort with us. So I want to start by saying that it's kind of a complicated thing to find out that you have a KIF1A diagnosis, because at first it seems like you found the gene, we have one disorder, it's KIF1A associated neurological disorder. And then you find out that you have one of maybe hundreds of mutations, and many people have very different mutations, they manifest differently, and so then it can almost feel like, oh, there's 100 different kinds of CAND. But in the middle, a primary question is, how does the mutation affect the protein? Because that has consequences for the body, for the health of the individual, for questions like progression. And it's also really important because when we model CAND in cells and mouse models, we want to make sure that we're representing as wide a swath of the patient population as we can. And somebody who's really been tackling this is Dr. Jayne Aiken. I think Jayne has a familiar story where she started getting into KIF1A, and immediately KIF1A.org scooped her up and said, hey, come work with us. And in the time since, she's provided a huge amount of help and information and perform research that's really meaningful, including informing the iPSC cell lines that we wanted to make so that we could study this disease in an equitable way that was still manageable and tractable. I'm also very excited to say that Dr. Aiken has now received a new position at Colorado State University, and she is going to be beginning a research program all about CAND. And so this is another example. Scoop them up early, get them in early, and they will thrive and grow with us. And so we're so grateful to Dr. Aiken for all the work that she's done, and I'm excited to hear a little bit more about heterogeneity and CAND.
All right, thanks, Dylan. And what he didn't say is I've actually known Dylan because we were graduate students together. And so I remember I came into CAND, and then when they scooped Dylan up, because he's been amazing, I was like, yes, more great friends, all part of the community. Yes, for those of you I haven't been able to meet personally yet, my name is Jayne. I'm one of the researchers that studies KIF1A. I don't know if we can grab my slides up. Oh, yeah, I don't have the clicker. No worries.
There it is.
Oh, haha. I have the power. All right. And as Dylan mentioned, today I'm going to be talking about heterogeneity in KIF1A protein and cellular function. And just as for an outline, today I get to talk to you about the exciting cell biology that KIF1A is performing within neurons. And so we'll start with a little introduction about motors and microtubules, shift into understanding how KIF1A mutations can cause changes to protein function, and how the change in protein function ultimately leads to different cellular consequences. So I like to start off talking about my favorite system in the cell, which is long-distance microtubule-based transport. And so we've heard a lot over the last day and a half about the brain and the spinal cord, but it's really important to remember that this system is made up of billions of neurons that are all linked to each other through cellular connections called synapses. And these neurons work together and send messages to, allow us to think and move. And inside of these neurons, we've seen this video before, KIF1A is performing a really important function to move different cargos throughout the neuron. And like Wendy, Dr. Chung mentioned yesterday, I also really like this analogy of KIF1A as a freight train on a track. Part of the reason why I love it so much is because it helps us contextualize that in addition to the train, the tracks are really important. Because without train tracks, your train's not getting anywhere very fast. And so in a cell, these train tracks are called microtubules. And these are long filaments that are formed from smaller little pieces called tubulin heterodimers. And not only are they important as acting as the train tracks, they also provide support for your neurons to extend these long arms that allow them to connect to their partners. And so here we go. You can see that KIF1A carries or walks along the microtubule in a specific direction. And this is another video, clip from that earlier video, which you'd seen kinesin walking before. But I also like showing this one, which shows a microtubule and how dynamic it can be. It can grow and it can shrink. And so this is really important because unlike a train going along a set track, in a neuron, your KIF1A trains actually have to deal with a train track that is being remodeled, much like if you were to add an excited kid or two to the train track set. And so how these trains respond to their tracks and help build that neuron is a really important fundamental question in how the brain develops and how it goes wrong in KIF1A associated neurological disorder. I also wanted to take a moment here to address a great point that was made earlier. So everyone in this room is really personally connected to CAND, but it's important to remember that everyone in the world has KIF1A motors right this moment walking along microtubules in their brains, and it's helping us be who we are. And so this question, by studying KIF1A and these specific mutations, we're helping inform what it means to be a human and how our nervous system works. So this research is not just important for this very specific, powerful community, but it actually can be broadly applied to all kinds of neurological disorders, from other developmental disorders and even neurodegeneration. And so in my lab, I'm really excited to be a part of this community and leverage this really great information that we've collected to understand how KIF1A motors and their microtubule tracks help build and maintain the nervous system. And so that brings me to the next part of my talk, where we're thinking about how KIF1A mutations can specifically alter different protein functions. And so, yes, here we are. We all are familiar that KIF1A mutations can lead to neurological disorders that present in different ways. And in fact, one KIF1A warrior can have very different symptoms than a KIF1A warrior across the table from them. And part of the reason why there are these differences, or our sciencey way of saying that, is this heterogeneity, is because you can have mutations to different parts of the KIF1A protein. And we've seen schematics like this before, showing where they occur. But another way to classify the different mutations is by how they are predicted to alter protein function. And so Karis and I have made this schematic by scouring previously published data and inferring how different specific mutations are predicted to impact the protein function. And you can see up there in the corner, there's still a lot of these mutations that haven't been investigated. They're in the unknown category. And one of the future directions of my lab is getting those off of that unknown list and put into their specific functional brackets. And just to walk us through how different mutations can alter KIF1A protein function, I have this little animation. So normally, another sciencey phrase is called wild type. So a non-mutated KIF1A protein has to dimerize, so that it has two little feet, it has to bind to its cargo and walk. And specific mutations can change its function in different ways. Some of them can lead to a loss of its normal function. So you can cause a shortening of the protein. It could cause it to not be able to dimerize with its partner. It might not be able to bind to its cargo. It might not be able to bind to the microtubule. All of those things would stop it from carrying its cargo where it needs to go. Sometimes it can bind to its cargo and the microtubule, but it leads to a movement defect, which we call hypoactive. Hypo is less. Alternatively, different mutations can be gain of function. So it's doing something beyond what the normal protein is supposed to do. So an example of this is it could be hyperactive, too active. It can go too far, too fast. Alternatively, it can be what we call Riger bound to the microtubule. So it can bind to the microtubule and get stuck. And this in particular mutation is quite severe and leads to some of the most dramatic symptoms that are apparent in the community. Another type of mutation can cause the motor to bind too tightly. So all those would be examples of it performing a function beyond the normal. And so this takes me to the last part of my talk, where we're really interested in figuring out how these specific changes to the protein and how it works leads to differences in the cellular outcome. And so, as Dylan mentioned before, myself and others in the lab were really excited to be a part of the team picking specific mutations to model an iPSC line that was developed by Jackson Labs. And in this initial study that I'll talk to you today, we picked out mutations that represent a broader class of changes to the protein. So we can figure out what happens to a neuron when it completely lacks KIF1A. So like the C92 stop, it won't have any full-length KIF1A protein. What happens when all of those motors are not going fast enough or far enough, that hypoactive, like the P305L, or alternatively, what happens when they go too far, too fast, the hyperactive R350G. And it's important to mention here that for this initial study, we used homozygous mutations. So all of the KIF1A in the cell has these changes. And that's actually different than what's happening in most of our patients, which are heterozygous, so they have one normal copy. But we wanted to use this as a clean slate to determine what happens in the cellular context when all of the KIF1A is altered in this way. And so we can take that iPSC resource that was generated and induce their differentiation into human neurons, which is really exciting. And this technology has rapidly progressed. I mean, even over the course of my postdoc the last 6 years, where we can take iPSCs and turn them into specific types of neurons and really get an understanding of the human cell biology that's happening with this system. For this study, we turn them into cortical-like neurons, so the neurons that are in that squiggly part of your brain. And as I mentioned, Karis and I have worked closely on this project to understand how different classes of mutations change neuron function. And what we found is that they can have lots of different changes, but some of the that I'll talk to you today is that different types of mutations alter the way that neurons are able to reach out those arms that are important for connecting to one another. So these loss of functions where you don't have KIF1A or KIF1A is not walking as well actually lead to delay in that extension, whereas the hyperactive seems to be able to send those out relatively normally. We also were really interested in looking at the very established known role of KIF1A in trafficking synaptic cargos. So here's the title page of the paper that first identified KIF1A in 1995. And even from that moment, we knew that KIF1A was particularly important for sending out these proteins that are essential for setting up and maintaining those synaptic connections between cells. And so we are able to look at what happens in live neurons to these synaptic cargos by you've got all these little things zooming around the cell. And I can create charts like the one that appeared below it, where you can see how fast things are going. So these diagonal lines are cargos that are moving quickly. The up and down lines are where things are just sitting static, they're stopped. And we can track how these different changes to KIF1A function change the way those cargos move. And so here's an example of those charts that I can create for these different classes of mutation. And you can tell by eye that they look a little bit different, but you can appreciate that more if we quantify the data, so if we plot, the specific values. And we're excited to see that when you have the hypoactive mutant, which is in orange there, the synaptic cargos, there's fewer of them and they're moving slower, which is what we would have predicted based on like how it was classified. As well as the hyperactive mutation, you're seeing more of these cargos out in the axon and they're moving faster. And so interestingly, the one where you don't have any KIF1A in the neuron, you actually get cargos, the right number, and they're moving about the right speed. But if you look at the intensity or like how bright it is, it kind of gives you some readout of how big these cargos are, and they're much smaller. So it seems like KIF1A is important for being able to carry the right size cargos, even though there are other kinesin motors in the system that are able to do it. And so with that synaptic trafficking data, we were really interested in seeing how that changes where these cargoes are distributed throughout the cell. So normally these are made in the cell body, the little round part, and they rely on KIF1A and other motors to carry them out of the soma. And so you can see here with the wild type condition, you've got some of them sitting in the soma ready to be distributed. But when you have a loss of function KIF1A mutation, you see more of it built up there. The trains are not taking this stuff out of the city, right? Alternatively, when you have a hyperactive, you actually get less of it in the soma. More of it is getting distributed, getting sent out. If we move along down the axon, you can see that the opposite is true, where you get less of this stuff in the axon and these loss of functions and more in the gain of function. And if we force our neurons to form presynaptic zones by adding a postsynaptic marker to another cell, you can see that they're not as good at forming these presynapses, even when we tell them to do it when you have the loss of function. And with the hyperactive one, you actually see a little bit more accumulating at these sites. I want to take a little bit of a shift of gears from the motor side to focus on the microtubules now. So we can't directly visualize microtubule filaments, but we can see where they're growing in neurons by tracking where the ends are being added. What you see is they look like comets shooting across the night sky. That's when we call them microtubule comets, because you can see it's like a shooting star of new microtubule growth. And just to bring this back to that original analogy, this is like new train tracks being built. And then remember, they can also go the opposite way where they can be disassembled as well. And so where microtubule ends are positioned in the neuron is actually really important, because unlike the schematic that you may have seen before and that I have been showing you this whole talk, synapses don't just occur at the end of this arm. And in fact, the majority of synapses in our brains right now actually occur all along the length of these along the length of these neuronal extensions. And so understanding, like, how does the cell know that it was supposed to create a synapse there? Or how does it know that it has to keep delivering stuff? How do the trains know to keep delivering things to these positions was a really important question. And so what we found through a number of published papers is that these specific synaptic sites are actually hotspots for microtubule growth and shrinkage. And that's really important in terms of KIF1A because this kinesin motor is a marathon runner, right? So it's gonna keep walking along the microtubule until it reaches an end. And so the positioning of these ends are important and act as like a stop sign. Stop here, deliver things here. And so I was interested in seeing how specific KIF1A mutations respond to these important train track cues. And so here you can see the green in this case now are those new microtubule growth events. The pink is where synaptic cargoes are accumulated. And we found that with our KIF1A lines, we see that same pattern of reoccurrent microtubule growth corresponding to where presynaptic cargoes are being accumulated. However, in the case of the hyperactive mutant there at the end, the R350G one in blue, even though those cues are there and you have this like stop sign microtubule cue there, the cargoes are not pausing. So there's a lot of cargo getting out there, but it's disregarding its important signs of where to go, which we thought it's really interesting, and it's acting in a way that's different than other KIF1A mutations. And so this is quantified here. We can see there's generally no change in how the microtubules are set up, but what that means is that when you have a hyperactive KIF1A mutation, there's fewer of these presynaptic accumulations along the axon. And actually, interestingly, there's a trend towards too many of them in the hypoactive, the P153G. So that's something that we're interested in following up on, this difference between the two different KIF1A classes. And so this cellular data was all really exciting, but what does that mean functionally for a cell? So a neuron's job, it doesn't just exist on its own. It connects to its partners and it receives and sends signals. And so to look at that function, I turned to a really exciting new technology in the field called multi-electrode array technology. And basically, that's an exciting way where cell biologists like me can grow cells on these electrode plates and record their synaptic activity. And so here's another video. This one plays. This one isn't even as exciting as the other ones. But anyway, what you can see is there are these little spikes happening in real time, and you can give this as a readout for how active that neuron is, how much it's firing. And so we can do this over time in these different cultures with these different KIF1A mutations. You can see in the black line, you get this nice increase in synaptic activity as the neurons are growing and maturing. But interestingly, even though that hyperactive one, which appeared to be, doing okay in these different cellular assays, it kind of goes up and then plateaus. And you see this, an even more significant decrease in the loss of function. So even though what's happening inside the cell is different for these different classes of mutations, they're converging on a shared synaptic output phenotype, where they're not able to pass on synaptic messages as well as a cell that has normal KIF1A. And so I just like to end with a little schematic of how these different classes of mutations can lead to different changes in neuron development. And we're really excited to continue to explore these other classes as well. This was an initial study where as Dylan mentioned, I'm starting my own lab. I'm able to take on this project and continue to look at how these different classes alter neurodevelopment and really have the ability to test in a high throughput manner how different therapeutic strategies could be beneficial for one class, whereas another therapy strategy could be more beneficial for another, which I think will have a really big impact for our CANT community. And with that, I just want to thank the lab that I did all this work in was at the University of Pennsylvania in the lab of Erica Holzbauer. Karis, who's here at the conference, is an awesome grad student who's done a lot of this work with me. And also a plug for my new lab at Colorado State University, as well as the funding. So thank you all very much for your attention.
Do we want to start with an online or do you want to go in room?
Yeah, sounds great.
All right. We have several good questions online. The first one is, does the overexpression of other KIF proteins, for example, KIF1B, restore the phenotype?
That's a great question, and we're actually currently working on doing that. Well, so there's a kind of multiple part here. We found that changing by mutating KIF1A, the cell itself isn't overexpressing any one of these KIF1A or an alternative kinesin motor. But we are interested in can we add back in either KIF1A or an alternative motor and rescue the phenotype. I think that is an especially attractive strategy for the C92 stop where you just don't have any KIF1A. And it looks like there's an alternative kinesin that's able to somewhat rescue, but not fully for different contexts. So that's a really great question, and we're currently exploring that direction.
Oh, great. While I'm walking over there, I had another question about the large list of unknown variants. Yeah. Obviously, we all want to reduce that list, but what does that process actually look like? What does it look like to start getting these into the right buckets?
Right. So luckily, it's not as daunting as you might imagine. So there are certain cellular assays that will tell you right away, does it bind to a microtubule? If it's on the microtubule, is it able to move as well as it should? And so that's an exciting project that I'm floating by a number of students right now, where there's fairly efficient genetic techniques to introduce new mutations, and then we can just pop them into cells and see where they fall. And then that part is relatively easy. The next step is harder. So modeling the specific mutations in the iPSCs is more challenging, which is why we were so lucky that Jackson Lab was able to create this resource. But that is actually challenging as well. So we did a lot of this work on a Riger mutation as well, because we were very interested in that. only to find a year later from Jackson that the editing wasn't what they thought, and it was actually like a mixed population, so that all the data we generated from that wasn't as informative as we'd hoped. So being able to partner with people with different expertise, which has been a theme of this conference and of this community, is really important. So, I can fill in some of the blanks, but then being able to rely on someone with a different expertise to get resources or to share my resources with them is really important for a more complete picture.
Our brains need fuel to operate, obviously. Have you looked at the fuel that powers the KIF1A protein and the neurons, and possibly looked at overfeeding those proteins to make them work faster or slower or more efficiently?
That's a really great perspective. So what you're alluding to is like the power of a cell, and of KIF1A specifically, is ATP, right? Which is generated through a number of different processes. Actually, interestingly, I don't know how many people know, a lot of times we think of mitochondria as generating the ATP that we need for a cell. Long distance trafficking actually relies on a different source of ATP. So, and a lot of times that's intricately linked to its environment and like other cells, so like astrocytes and glia cells. So I think that's a really interesting question of figuring out like how the cellular environment, how its fuel source is impacting the proteins and their ability to move, and whether altering that, and then taking that even further translationally, if you can do a supplement or a diet or something. I think that's a really interesting perspective that I would not have thought of on my own, which is why it's great to present to a wide audience. So thank you very much for that comment.
We'll take 2 questions from online next. How long does it take for kinesin proteins to transport their cargo down the neurons? Seconds, hours, days?
Great question. So what I presented here is that a KIF1A motor can walk approximately 4 microns per second, which is really fast when you think of motors, but it's hard for our brains to actually comprehend that because it's a very small distance. I would need to calculate out for like the length of, a typical axon in the brain, which is going to be a lot shorter than a peripheral axon that goes from your spinal cord to your toe. But I actually will do that calculation so I can have it in my next presentation, because it's fun to think about that. But it is on the scale. It's not seconds. It's going to take a lot longer than that. And in fact, most of the time, a single KIF1A motor is not going to go from the cell soma all the way to the end. There's a lot of dynamics in this situation where motors pop on, pop off. There's regulation in the cell that degrades and makes new proteins. And so you can think of it more as teams of these motors being responsible for carrying a specific cargo out to where it needs to go.
I want to follow up on this because I know the answer to this, and the answer is that it's kind of cool that we don't know. And this is, I think, something that's important to remember in biology and in these projects and these questions we're asking is we know how fast these motors walk, and we know how fast they need to walk for our bodies to function. And when you do the math, it doesn't make sense. Like there's no physics way that they can be walking as fast as they are with the information that we have, which doesn't tell us that they're not walking. They obviously are, because I can kick my leg and it's KIF1A's walking and tell my leg to do that, but it tells us that there's some piece of the puzzle that we're missing to help us understand and contextualize the information that we have. I want to save you a bunch of math, but we can talk more about it later.
Just the idea of like, if you had a single kinesin motor walking for this specific distance, this is how long it would take. But that's, like, as I mentioned, that's not actually what's happening biologically, but it would be interesting to see at the rate that it moves, how fast could it get there?
The next online question I will read off because I have no idea what it means. Does the activation of lysosomal exocytosis restore the secretion of KIF1A dependent cargo?
So lysosomal and secrete.
Does the activation of lysosomal exocytosis restore the secretion of KIF1A dependent cargo?
So KIF1A dependent cargo secretion could be alluding to synaptic release. And synaptic release is tied into a lot of other cellular processes. So lysosomes that they mentioned are like the trash cans of the cell. There's actually another trash can of the cell called autophagosomes, and that's actually really intricately tied to synaptic activity. The long story short to that question, I'm not sure how lysosome dependent secretion would be directly impacting the secretion of the cargoes delivered by KIF1A and whether those pathways are talking to each other. But it is very possible, and we haven't looked.
And we'll go to an in-room question next.
Thank you for this wonderful mechanistic dissection. My question is, do you have any plans to look at how the heterodimers of KIF1A wild type with some of these different loss of function and gain of function mutations functionally impact the cargo transport within the cells?
Absolutely. That's a really important next step. So now that we've understood what it means when a cell has all of its KIF1A altered, what does it mean, like we see in the patients, when half of the KIF1A is altered? And how does that change these cellular outcomes? Does it make it worse because the stoichiometry of you had some good copies, but now they might be working worse because they've got the mutant? How does that change these cellular outcomes? And that's a really important next step that we're actively working on.
All right, thank you, Jayne.
Thanks.